showing, I think it leads perfectly my talk after yours. These are my disclosures, and we know that RSV, it's a leading cause of hospitalization in infants in the developed world, and in the developing world is the second cause of mortality in this age group. We also know that the majority of infants that are hospitalized with RSV are healthy. Of those, around 15 to 20% will require intensive care unit admission, so they're sick enough to be admitted to the ICU. And although we know epidemiologic factors that are associated with disease severity, they do not completely explain the huge variability that we see in this disease severity in children. In vitro and animal studies suggest that RSV may enhance nasopharyngeal colonization with some bacteria, facilitating bacterial infections, and that also there's more and more data coming along suggesting that specific nasopharyngeal microbiome composition is associated with increased risk of respiratory infections and also RSV, and as well as the development of asthma. Years ago, 7 years ago or so, we decided to look not at the whole microbiome initially. We decided to look at potentially pathogenic bacteria that have been associated with more severe disease. We did this study by culture in hospitalized patients. They were all inpatients. So we set up enhanced cultures to detect for potentially pathogenic bacteria: Pneumococcus, Moraxella, Staph aureus, and Haemophilus. We enrolled in the study during one respiratory season more than 136 patients. However, when we were about to analyze the data, around 50% had received antibiotics, and these patients were enrolled within 24 hours of admission, so cultures were negative. So we ended up with analyzing a cohort of 72 patients with RSV that were hospitalized and not exposed to antibiotics, and in parallel to determine if the prevalence of bacterial colonization was higher in RSV patients compared to control, we enrolled a cohort of healthy age-matched control. So we saw first in the RSV cohort that actually 81% were colonized with one of these potentially pathogenic bacteria compared to 65% in the healthy age-matched control group. But not only that, when we looked at the proportion of bacterial colonization with those bacteria, we saw that healthy controls were mostly colonized with gram-positives in dark blue Staph and in light blue Pneumococcus. But look at the proportion of colonization in infants with RSV was different. There was a higher proportion of colonization with gram-negatives, Moraxella, but also Haemophilus, and also polymicrobial infections. So we saw that in healthy infants, these are not high-risk infants, so healthy infants hospitalized with RSV infections were highly colonized with these potentially pathogenic bacteria, and that the profile of bacterial colonization was different compared to matched controls. So the next question was, is this doing anything? Is this a passive phenomenon? Is this associated with more severe disease? So what we saw first, we did nasal washes in all these patients, that these RSV patients colonized with any of these potentially pathogenic bacteria. They had more white blood cells in the nasal wash, it makes sense, but also at the systemic level, they had a greater proportion of neutrophils in the blood. We measured in a subset of these patients in whom we collected serum some cytokines, innate immunity cytokines, IL-6, IL-8, and TNF-alpha that have been associated with actually both RSV and bacterial infections. So here in black represents, you see that the groups were small, so we need to take this with a grain of salt, but it really laid the foundation for the future studies that we did. But in black are represented the gram-negatives, in light gray the gram-positive bacteria, in white patients with RSV colonized with regular respiratory flora, and these are the age-matched controls. So what we saw in the serum of these RSV-infected babies compared to controls, that those colonized with gram-negative bacteria had higher concentrations of both IL-6 and IL-8. This was done by culture. We enrolled a subsequent cohort of patients looking at these bacteria by PCR, but we wanted to go one step further and decided to look at the whole microbiome. So this is the proposed model. There is a synergistic interaction between RSV infection, disease severity, the host immune response, and the nasopharyngeal microbiota cluster, with the hypothesis being that RSV disease severity is influenced by modulation of the host immune response by the local bacterial ecosystem. So we enriched our cohort and enrolled a further cohort of patients. They were previously healthy children with RSV infection, hospitalized, and also enrolled in the outpatient setting. We enrolled a cohort of age-matched control, perform a nasopharyngeal microbiome analysis targeting the sequencing, the 16S ribosomal RNA of the bacteria, and also in parallel to understand better the meaning of this colonization, we did transcriptional profiling in the blood of these infants. And of course, to integrate all this multidimensional data, we did multivariable analysis. So we enrolled 132 participants, 26 healthy age-matched controls, and these are really hard to enroll to obtain blood from these healthy babies, and 106 children with RSV infection. The majority were inpatients. 84 out of those, 1/3 required PICU admission, so they were sick enough, and the rest were in the regular ward. And we enrolled 22 outpatients. So with Debbie Bogart, we decided to look at this, the whole microbiome, and you'll see and how it is associated with both with severity and with the host immune response. We did first an unsupervised cluster analysis, looking at the whole microbiome in the whole cohort, in RSV patients and controls. And we found 5 microbiota clusters: Strep pneumococcus, Haemophilus, Moraxella, Corynebacterium, and Staph. And in this unsupervised cluster analysis, the Streptococcus and Haemophilus, those clusters were enriched with RSV patients. Is this bacterial colonization associated with any differences in severity? So doing NMDS bacterial clustering is like principal component analysis. You integrate the microbiome data with clinical parameters. We found actually with Wouter and Debbie that these two clusters, the Haemophilus and Streptococcus, were actually prevalent, much more common in patients, so patients with more severe disease, compared to these other 3 clusters, the Moraxella, the Corynebacterium dolosigranulum, and the Staphylococcus, that were clustering with the outpatients. These inpatients were younger and also required more antibiotic therapy, meaning that they were probably sicker. So doing this unsupervised cluster analysis and integrating the clinical data, we were able to separate clusters of bacteria that were associated with a different disease severity phenotype. But we wanted to understand this better, and to make things a little bit more complex, we were asking, is this a passive phenomenon? How can we understand the role at the immune system level of bacterial colonization? So we decided to combine the microbiome with the host transcriptional responses. And we did microarray analysis in the blood of these babies. So doing first pathway analysis, this is ingenuity, we found and confirmed what we've seen before in children with RSV, that there was a common pathway dominated by overexpression of interferon-related genes, but then depending on the bacterial colonization cluster, there were different pathways activated. The Streptococcus and Haemophilus was mostly dominated with IL-8, IL-17 pathway activation, and these pathways were not present in those infants with RSV and yet colonized with this bacteria, Corynebacterium, Moraxella, or Staph. So they were associated with less disease, and the pathways activated at the systemic level were different. Another way of looking at the data, we decided to do another type of analysis using David that identify a subset of cytokines, innate immunity cytokines related to IL-8 and TNF-alpha. Here each column represents the transcriptional profile of a patient, and its row a transcript. In green represented the healthy controls, blue Haemophilus cluster, Streptococcus cluster, Corynebacterium, Moraxella, and Staph. Visually, you can see that these 2 clusters had greater activation of overexpression of all these transcripts. And also looking at the modular type of analysis, another pathway analysis, we saw that as expected, all of these patients infected with RSV, independent of the colonization pattern, had overexpression of interferon. The proportion of genes related to inflammation and to neutrophils were significantly higher in patients with RSV and colonized with Haemophilus or with Streptococcus. So in summary, we've seen that nasopharyngeal bacterial colonization with potentially pathogenic bacteria is common in children with RSV, that patients with more severe disease, so in patients have a different pattern of nasopharyngeal colonization, that transcriptional immune profiles in infants with RSV bronchiolitis are different according to the nasopharyngeal microbiome profile that is associated with the severity of the disease. So infants with RSV colonized with Haemophilus and with the Streptococcus were mostly in patients and had a different systemic immune response. So possibly viral bacterial interactions in the upper respiratory tract may open a new approach, a new way to analyze RSV disease severity in this population. And to finish, I would like to thank all my colleagues and collaborators, especially Ravio. We share a lab. We've been working together for 15 years, and David Bogart from Utrecht University. Thank you very much.
Very good indeed. Can I just ask, what's the effective time after onset of disease? Is that a factor that you take into account?
We adjusted for time. These patients are usually enrolled within 3-5 days of symptoms, but we don't have dynamic data just yet. So we don't know how the microbiome composition will change over time. So far, when we adjusted for age, we didn't see age. For timing, we didn't see that much differences.
And the transcriptomics?
In the transcriptomics, we did some initial analysis and compared children that have been enrolled within 3 days of symptoms, 5 days of symptoms, and in acute diseases, usually the signal is so overwhelming that somehow takes over these differences.
Great. I think
we're analyzing now a subset of inpatients versus outpatients, which is key, which is going to give us a lot of information.
Ross Smith, University College London. You seem to imply that the microbiome determines severity of RSV infection. Don't you think it more likely that the damage from severe infection in the airways alters the microbiome as a secondary phenomenon?
Well, I hope I wasn't that dogmatic. I think it's a factor that we need to start thinking about that I would have never thought about years ago. I don't think it's the only factor. I think it may play a role, but I don't think it's the only main. It's not playing a role.
Jürgen Schwarze, Edinburgh. I guess my question is very similar coming from the other end. And so, I mean, to me, the critical question is this pre-existing microbiome or not? Do you know from any other studies, because you don't seem to have the data, whether within 3 to 4 days of a viral infection, a microbiome can switch completely? Or, so basically, what's your take? Is this pre-existing, what you see 3 days into an infection? Or is that potentially done by the virus?
Well, to answer that question, we would need to have a birth cohort study. There are a couple of them. Most of these studies have been done in patients at high risk for developing asthma. In those studies, it was very interesting because it matched with what we found. When these patients are healthy and they have sequential samples from birth, they are colonized with Corynebacterium, with Staphylococcus and Moraxella. However, when they have an acute respiratory infection, the proportion of Haemophilus or Streptococcus increases significantly. So there seems to be an association that we need to understand better. But yeah, we are actually doing a birth cohort, studying a birth cohort, and hopefully we'll have better answers.
Last question.
Steve Cunningham from Edinburgh. You excluded in your initial study the patients that had received antibiotics, but they were probably the interesting group, weren't they? Because presumably it's an easy infection to treat. They would have had a better course, a simpler course.
Well, I didn't show all the data, but in that paper that was published, we actually compared, because we were wondering, patients that had received antibiotics versus others that didn't, and those that had received antibiotics were sicker. They were longer in the hospital and they required oxygen for a longer time. For the microbiome analysis, all patients were included because we done the analysis by pyrosequencing, and antibiotic didn't seem to play such a big role. But in the first study, we did compare these patients and they were sicker.
Great. Well, thank you very much indeed.